So good afternoon, everyone. This is David Collins, the Chief Commercial Officer from Aptamer Group. We have a good number of people connected into the webinar. Hopefully a few more will arrive, so I'll just say a few words of introduction. Hopefully the technical aspects are all working and that you can hear my voice clearly and see a good image on the screen. We are monitoring it and it looks good, so hopefully we'll have an interesting and useful session for you, and the webinar will have some value. So I just want to welcome everybody that has connected to hear us talk. This is the first Aptamer Group webinar in a series that we've got planned. We've been overwhelmed by the excellent response. We've had nearly 100 people registered globally to hear about Aptamers, so that's very pleasing for us. And in this integrated series of 3 presentations, what we'll cover today is Aptamer selection, and then in the follow-on presentations we'll move on to their applications and uses. We are in fact planning a 4th webinar. The date hasn't been set yet, but that's for the first quarter next year, when we will focus on cell targets. I'm pleased to introduce my colleague, the presenter for this series, Dr. David Bunker. He's the Chief Technical Officer here at Aptamer Group and a co-founder of the company. He's been involved with Aptamer science for more than 18 years now, starting in academia, but then emerging from academia to set up the company in 2008. And during his career, he has selected more than 200 Aptamers against a very wide range of target types. So that's what we'll hear about over this series of presentations. At the end of the presentation, there'll be a question and answer session. We will be happy to receive these typed questions from the link on the YouTube page that this webinar is being broadcast on. And there's also a form on our website. So without further ado, I'll hand you over to my colleague, David Bunker, to continue with the presentation. Thank you for joining us.
Thanks, David. And thanks to everyone for joining us on this first of our seminars, and we'll get started. So as David said, this is going to be the first of 3 seminars. So the next of which is due for a broadcast on the 25th of October later this year. And then just over a month later, we'll have another one on applications of small molecule targeting Aptamers. So just there for dates for your diaries, and hopefully we'll see you there. So the first thing I would like to go through with you is essentially how the customer interacts with Aptamer Group. How do we work? Why are we different from other Aptamer providers? And what we'd like to do with this seminar series is get people familiar with the idea of Aptamers. What can we do with them? How can they integrate into our science? And how do we engage and how do we access Aptamers? Well, our process is actually broken down into several key stages, and we like to think of these as essentially milestones along the road to having a successful Aptamer. And the first of these is a step that we've actually broken out of our standard process quite recently and made this upfront what we refer to as an Aptamer feasibility study. So that's a short piece of work taking only 2 to 3 weeks, absolute maximum, where we will engage with the customer and take your target of interest, whatever that happens to be, and we'll perform some simple studies to say, is your target actually amenable to Aptamer selection? And the reason we've done that is a lot of people come to us and say, well, I'm not really familiar with the technology. Is it something that's going to be good for me? Is it something that my target will even work with? So we've broken that out as our first milestone to say to a customer, yes, your target is good for Aptamer selection. We have X confidence that we will be able to generate you an Aptamer. So that's a simple go, no-go decision, and we proceed from there with the Aptamer selection in full. So that comes to the next point on this slide, the Aptamer selection itself. So we have several different processes in-house depending on the nature of your target. So our small molecule selection process is different from our protein target selection process, which is indeed different again from our targets of complex natures like cells or tissues or even FFP sections. Essentially, whatever you can think of, we can probably generate you an aptamer against it. We just have different processes for that. Next stage in our process, once we've got an aptamer population, is to then identify individual aptamers. So to use antibody terminology, we'll refer to those as monoclonal aptamers, and screen those for binding to your targets. So that's what's going to give you your individual sequence at the end. Once we've done that, not all customers need this final step of minimal fragment identification, but it is part of our process where necessary. So that's essentially taking the full aptamer sequence and finding the bare bones, as it were, the minimal fragment that's required for function. So the reason you might want to do that is to bring down production costs. So if you're integrating your aptamer, for example, into a diagnostic device or even a therapeutic, you want the smallest fragment that's possible to keep down production costs and make things easier for yourself. So that's the service we also offer. In addition to aptamer selection, we also help customers to integrate the aptamers into an array of diagnostic platforms, some of which I will mention very briefly at the end of this seminar. Others we'll touch on in more detail in the next webinar. So that can be anything from ELISAs, lateral flows, anything like that. We also help with purification column development through our Aptabind service, which is again using some of the unique properties of aptamers to purify a protein. And very briefly at the end, we're looking also in one of the seminars next year about using aptamers in biomarker discovery. So that's just to give you a flavor of what's coming up. But it's always very important to note that we always start, as we say here, start with the end in mind. So what we mean by that is the first question we will ask you is not what is your target, it's what do you want this aptamer for? How do you need it to work, and what do you intend to do with it? So we start with the end in mind. So to come to the start, what is an aptamer? Well, in simplest terms, the easiest way to think of an aptamer is a synthetic piece of DNA or RNA with modified nucleotides present that's been isolated to do a particular job, to recognize a target of interest. So the easiest way to think of them is the nucleic acid equivalent of an antibody. And what you can see in the little cartoons here is the structure of 3 different aptamers binding to their respective targets. So the simplest representation on the right-hand side there, you can see the aptamer in purple binding to a protein shown in orange, and that interaction is very much like you would expect to see with an antibody. So a compatible face on the aptamer interacting with the protein. The other 2 targets, however, are small molecules. And in this case, aptamers are actually quite different in the way they work. So rather than just creating a suitable binding face to sit alongside the protein, what we would tend to do with small molecules is actually fold around the small molecule and almost form a cage. Now, obviously, this is something that antibodies can't do. So this is one reason that we say aptamers are actually a lot better for small molecule targets than antibodies can ever be, due to this ability to cage the molecule. So as I mentioned, aptamers are synthetic pieces of DNA. So what I mean by that is obviously they're created entirely in a lab. So all in vitro processes, there's no animal use in aptamer selection whatsoever. And that gives us a number of advantages. Firstly, it means we can work with almost any target type. So you can imagine it's very difficult to try and elicit an immune response against a paraffin-embedded tissue section, for example. Well, we work in a lab, so we can work with tissue samples like that. We can also work with small molecules or proteins that might be inherently toxic, or just don't stimulate the immune system at all. So we really can work with a much broader range of targets than you might get from other technologies like antibodies. We're also not limited to a biological environment. So a scenario that we come across very, very often is customers come to us saying, I have an antibody that recognizes my target, but I've tried to convert it into an assay, and when I use it in a biological matrix, or a tissue sample, or let's say milk for an end application, that antibody just doesn't really work anymore because it's not designed to work in that environment. We can take that into account, starting with the end in mind, and say, right, we know that this aptamer needs to work in milk, so we will raise the aptamer in milk. We can also tune the specificity and cross-reactivity and build that into our selection process. So this is something that, again, is quite unique to aptamers. You can't really do this with antibodies without having to go through a long and arduous process of screening to find the cross-reactivity or specificity you need. We can build that in, and I'll show you how to do that in a moment. The end application, obviously, you're going to need to produce these aptamers, and this is, again, another big advantage for this technology. They're produced synthetically through very well known and very well understood chemistry, and batch scale production is now very well characterized. You can have an aptamer made on up to the kilogram scale if you need to. So that means that batch to batch variability comes right down, because you might say, well, I only need one batch for the lifetime of my project. The other point to remember with an aptamer is, at the end of the day, it is information. It's a sequence. So you can produce that in 20 years' time without having to worry about decay or degradation or death of a cell line or anything like that. You can just open up a file on a computer and have the same aptamer made that you made 20 years ago and know that it is going to work. The other point is the time for this process. So aptamer selection can be very, very quick. As I mentioned previously, our upfront screen to say, is it feasible, can be as little as a week. We say up to 3 maximum. And then the aptamer selection itself is typically between 8 and 12 weeks. If we get really lucky and you have a very good target, this can be as little as 4 weeks. So it's a very, very quick process when you compare it to something like antibody generation, which can take between 6 and 12 months. And as I mentioned before, ours is a staged process with milestones where we can keep track of the process at every step of the way and keep you up to date on your progress. So once you've got an aptamer, you can then turn it into virtually any sort of tool you like. So again, as I mentioned before, there's very routine chemistry for the production of aptamers. You can also have them made with routine chemistries on the ends of them. So for example, if you put a thiol on the end of your aptamer, you can then functionalize that aptamer with a whole range of different moieties. So you could put a fluorophore on, or a gold nanoparticle. You could attach it to a sensor surface, all using the same chemistry. So you can have one aptamer and use it in several different applications just by putting different things on the end of the aptamer. It's a very, very simple chemistry to modify your aptamers, which enables their use in a whole range of different applications. And some of those applications, just mentioned here very briefly on this slide, the point here is just to demonstrate that aptamers are really starting to find their use in every area of the life sciences. And we've actually challenged ourselves and some of our staff to try and come up with an application in biology where they didn't think an aptamer would be suitable, and we haven't found one yet. And so there's just a few examples here, but what you can also see is that by the little logos on the right hand side there, we've tried to position ourselves to address as many of these needs as possible. So, if you need an aptamer, we're pretty confident that we can help you with that. So, on to the actual process of generating an aptamer. So, many people are familiar with this process. If they've come across the term aptamer before, they've usually come across in vitro selection. I'll just walk you through this just to explain how it works and how our process is a little different from other people. So if we start right up at the top there with our aptamer library, represented by these little, they look like sweets to me, but they're DNA helices. So they represent our aptamer library, and the little jigsaw pieces in the middle represent the target. That can be absolutely anything, as I mentioned before, a cell, a protein, a small molecule, it doesn't really matter. Let those two molecules interact with each other under defined conditions. So we're now at the stage of the Aptamer target complexes. We have precise control over that. So as I mentioned before, we can do that in, let's say, milk for this example. So if customers after an Aptamer that recognizes a protein in milk, that's how we will work here. We then, once those complexes have formed, we move on to the partitioning of the bound and unbound species. So that's exactly what it sounds like. Anything that sticks to the Aptamer is retained, anything that doesn't stick is removed and washed away. And we can actually go one step further with that and say, if the Aptamer is designed to recognize one target but not recognize a homologue, shall we say, or something related, we can actually remove the non-binding species as well as the non-target binding species. So something, Aptamers that recognize something else, they all go down the sink. We're not interested in them. We then recover the bound sequences, usually through a denaturation process, and recover those Aptamers and amplify them up. We can actually build in additional steps here though. So part of our Aptabind process, generating Aptamers for protein purification, as well as being able to bind the target, it's very important that the Aptamers can also let go again. So we might do that by adjusting the pH or adjusting the salt conditions, for example. So we can actually build that into this process as well. So we have defined binding conditions, but also defined release conditions. And the Aptamer needs to function in both of those situations. So again, starting with the end in mind and tailoring this process with the goals of the customer in mind. So once we've recovered the Aptamers, we then amplify them, and essentially we end up with an enriched population of Aptamers and go around this process again with that new library. So essentially we're going around this process several times, refining the Aptamer population as we go until we get to a situation where we have the properties that the customer requires. So this is how the idealized Aptamer situation would work. So at the start of our selections, we have approximately 10 to the 14 to 10 to the 15 different Aptamer sequences in there. And as you can imagine, they're going to have a range of affinities from very, very good on the left-hand side at picomolar to pretty poor with millimolar affinities at the right. And the name of the game, as they say, here is to recover the high affinity species and not the low affinity without messing up the selections in the first instance. And what I mean by that is it's very, very easy, if you don't get the selection conditions right, to carry through a lot of very weak or non-specific interacting Aptamers that would have, you know, in this spectrum, the micromolar to millimolar affinity. If you do that, you generally find that you'll outcompete the high affinity Aptamers in the early rounds and they'll be lost from your population. And the problem is, once you've lost them, it's very, very difficult to get them back. So it's all about retaining the high affinity sequences through this process and giving them the greatest chance of being enriched and carried forward. And then, as you can see, as the process goes on through round 3 and into the latter rounds, your population distribution changes to something with picomolar to nanomolar affinities. So that's the goal of the project. So a very, very short summary of the Aptamer selection process there. And what I'm now going to do is give you a few examples of how the process differs for protein selection versus small molecule selections as the basis for the talks going forwards. And what I'll start with is our upfront screen. So as I mentioned before, one of the questions that we quite often get is, well, I don't know that my target is amenable to selection. Why would I want to take this forward? How do I get some proof of concept? So that's why we designed this panel of 3 experiments. So the first one, at the middle of the slide there, is essentially a simple stability study. So a lot of customers, this isn't necessary. If you have a clean in vitro application, you might not necessarily need this. But if you're going to be working with a complex matrix, let's say milk or a food extract again, you might want to understand, is the Aptamer going to survive long enough for my assay to be carried out? So what we have here is essentially a concentration series, a time course, sorry, of a non-modified Aptamer on the left and a modified Aptamer on the right, incubated at 37 degrees in a customer's target matrix. And you can see in this situation, the non-modified aptamers on the left are gradually degraded by something in this customer's target matrix, whereas the modified aptamers on the right are perfectly stable. There's no sign of degradation. So just by carrying out this very simple experiment, we can say to a customer, you need a modified aptamer, whereas this customer here doesn't need one. So that's your first port of call and first piece of information that you'll get. The next piece of information is critical to getting the aptamer selections to work properly. So this is all about finding the best binding conditions. So many people say to us, well, how do you know what's going to work for my system? And we actually carry out a screen to identify additives for their selection or salts that will promote an interaction between the aptamer and the customer's target. So what you can see in this simple graph here is the green bars representing aptamers binding to the customer target, and the red bars representing aptamer binding to the background matrix. And what we look for in this screen is conditions that promote most favorably the interaction with the target and not with the background. So essentially the highest green bar to red bar ratio. So in this situation here, we would say that buffer condition 5 is most favorable to promoting interactions with this customer's target. So then we've got the best chance of generating an aptamer in that buffer. The third piece of information that we get is an idea of the interaction between the customer's target and the aptamer library as it stands at the start of a selection. So not all targets are going to be really high affinity nucleic acid binding proteins, so they're going to have much, much lower affinities for the aptamer library than, say, a transcription factor, which is inherently nucleic acid binding. So it's important for us to understand that, and that will also give the customer some information to say, yes, your target can be made to bind nucleic acids, we just need to put it under these conditions, and then we'll be able to generate aptamers. So as I mentioned right at the start, this is a 1 to 3 week piece of work that will then give the customer confidence that we're able to generate them an aptamer. And that's a stop-go decision. If the answer to this is no, we can't generate you an aptamer, you haven't committed to a long process of aptamer selection that's not going to go anywhere. So that's a real differentiator for us. We give our customers that confidence right up front as soon as we can. Once we've done that, we then move on to the aptamer selection itself. So I'm going to start with the small molecule aptamer selection approach. And here we actually work a little bit flipped on its head compared to a standard aptamer selection. So with a lot of small molecules, it's not really possible to immobilize them onto a solid surface for traditional selection, because if you modify that small molecule, it's no longer the same target. You're not going to have the same surfaces available for interaction. So we actually immobilize the aptamers instead, and then look for a displacement effect with the target. And any aptamers that get displaced with buffer get washed away. Any aptamers that are then subsequently displaced with the target are recovered. They're your binding sequences, and they go through the next generation. So same as in a traditional selection, they'd be amplified, expanded, re-immobilized, and we go again with an enriched library. So it's a very straightforward process. It's just flipped on its head compared to standard selection. And you can see here, this process works very, very well. So anybody who's read aptamer literature is probably familiar with these sorts of graphs. You see that in the early rounds of selection, there's little difference between background and target elution. But as the selection takes off, as we get exponential enrichment, you can see there's a great increase in the target elution compared to background. So that's a good sign that exponential enrichment is happening. Typically from round 7 through 9, we see that. For the assays for small molecules, we carry out the assay in the same sort of situation on a sensor surface. So we immobilize the aptamer, wash it, and then elute it with the target. So an idealized data set is just shown at the top left-hand side there, just for representation purposes. And what you can see when we screen our clones on the top right is some of the aptamers that are, or some of the sequences that are in that population don't have very good affinity for the target. That's perfectly understandable. Not everything in there is going to be an aptamer, but you can see very clearly there that there are 2 very strong hits come out of that population. And when we go to characterize them further with the data set at the bottom, you can see that this target in red, you can see that interacting with its chemotherapeutic target and being displaced from the surface, whereas the drug metabolite and the negative control have very, very negligible effects. So what this shows us is that this individual aptamer is specific for its target, it doesn't recognize the metabolite, and it doesn't really have a non-specific matrix effect. So this is an example of a really well-performing aptamer. Proteins are a little different in the process, but you know, the end goal is the same. So what we very quickly discovered when we set up the company is that using a traditional approach of aptamer selection is not really the best way of doing this. And what I mean by that is people would generally look at a selection target, look at the process and say, right, that's my end goal, and I'm going to take this path, and that's how I'm going to get there. So that might be, we'll increase the stringency at round 4, and we'll do more washes at round 5, and then we'll hope for the best and see what we get. And you might get lucky. Sometimes that works. But what we found is more often than not, you might put too much selection pressure on because you don't really understand the nature of the target. So what we've done is turned the aptamer selection approach into a multiple branched approach. And I'll explain what that means here. So if you imagine we've done a round of selection, we're going into our next round of selection, and we want to increase the selection pressure. So make it more difficult for the aptamers to bind to the target, so only the highest affinity sequences are recovered. But we don't really know what's going to be the best that we can push this without washing all the aptamers away. So what we do is we do the selection in parallel 4 different ways. And we can do that because we use high throughput robotic systems for almost all of our aptamer selection processes. So we actually carry out each round of selection up to, well, in this example, 4 different ways. There are more that we can work with. And then we will recover the material from each of those 4 different selection attempts and say which of these has actually given us the best enrichment. So it might be that condition 3 gives us the greatest enrichment, condition 4 was too harsh. So we would then take condition 3, and that will become our starting point for the next round of selection, and we would branch again. So every round of selection that we do branches, and you then pick the best route to the end goal. So the fastest route to aptamer selection by choosing your selection pressures as you go in real time. And again, you can see an example here of how well that works. So the selection pressure here is introduced at round 2. This is why you see the drop after round 2. But then you can see that our enrichment takes off, and by round 7, we would say that's at a point where we can start looking for individual aptamers. And we can also monitor that interaction by a number of different assay formats. The most common one that we use is biolayer interferometry, which is very similar to surface plasmon resonance that people are generally familiar with. So you essentially just get a sensogram like this, showing enrichment of the aptamer population as we go. And as I mentioned, we always start with the end in mind, and we also screen by function with the end in mind. So again, in this case, this is one of our AptaBind projects where we're looking for aptamers that bind the target, but also let go of it. So in this situation, we immobilize the aptamer on the surface, because that's what you're going to do on a column, show that there's no specific interaction at all with the buffer matrix. But then as soon as we add the aptamer target in there, you can see a very strong interaction on the top 3 curves. Some dissociation during the wash, which is what you would expect. But then as soon as we introduce the elution conditions, all of the target is released, and then we can go around the process again. So again, it's about screening with an assay that's suitable to the end goal of the customer. And then briefly to mention cell-based targets, the process is pretty similar. So we actually break a cell selection down into 3 phases. Phase 1 is really looking for enrichment of a population of aptamers that bind to a cell in general. Phase 2 is then when we would introduce a counter selection. So in the example here, we were looking at for cancer-specific aptamers. So we would introduce at this point the healthy cell line to then start removing aptamers that bind to the healthy cells. And then in phase 3, we push for the most high affinity and high specificity aptamers that we can get. So you can see the population takes a really strong hit at the start of phase 3 because we really ramp up the selection pressure, and you can see it very quickly recovers there. An example of the sort of aptamers we get from that. So here is some confocal imaging that we did with the cancer cell lines across the top and the healthy cell line across the bottom, and the aptamer is labeled with a green fluorophore, and you can see very, very clearly there that this aptamer picks up the cancer cell lines but doesn't really recognize the healthy cell line at all. So very, very specific aptamers. So that's a very quick summary of the aptamer selection process, how we work with our customers, how we go forwards, and the types of targets that we can work on. And just to give you a flavor for the upcoming topics on the next few slides. So our next seminar is going to be on protein-based applications of aptamers. So I'll be talking about ELONAs, that's the ELISA equivalent with an aptamer, lateral flow developments, and I've touched briefly on our Aptabind protein purification platform as well. We'll be extending on that in a little more detail. And then later in the year, we'll be coming back to the aptamers against small molecules with a couple of case studies there of using aptamers in a simple fluorescence-based assay, and using aptamers, as you can see at the bottom, to recognize target in a complex matrix like plasma. And then, as we mentioned right at the start, planned for next year is an aptamer cell-based seminar where we'll be looking at processes like fluorescence microscopy, flow cytometry, and even patient stratification, biomarker discovery, and drug delivery. So, just in summary, an aptamer, in its simplest way of thinking, is a nucleic acid equivalent of an antibody, but they're selected entirely in vitro under customer-defined conditions, starting with the end goal in mind. They can then be modified to give them a range of functionalities depending on your end assay, and you can use them in virtually any application. Wherever you can use an antibody, or wherever you would like to use an antibody, you can use an aptamer. And at that point, I'd like to thank you for your attention. I know we've had some questions come through while I've been speaking, so we'll take the next few minutes just to try and answer a few of those, and we'll see how we go from there.
Well, thank you very much, Dave. That was excellent. I think we might have actually addressed some of the questions, but this might precipitate a bit of further discussion around them. There's two related ones that we received earlier. It was, how do you know which aptamers are the best? And I think that sort of ties in with this other question we've been asked. How do you consider the interference and counter-target effects? Okay. So I think some words about affinity and selectivity here would answer those questions.
Yep, no problem. So the counter-selection targets and interference from background, first of all. This again is why it's very, very important to understand your end goal. So you might say to us, for example, to use our milk example again, I want to be able to recognize an antibiotic in milk, which is great, that's fine. But what it's important to understand is your end assay. So you might say, well, if we just generated the aptamer against an antibiotic, that might work fine. But then you'd get a background interaction with other proteins, other things that are in the milk. So we need to make sure you don't have that situation carried through into your aptamer selection. So we would do the selections in a milk-based buffer, and if there were any related targets, any other antibiotics, for example, that you didn't want to recognize, we would include those as counter-selection targets and remove them. In terms of how do we know which aptamer is best, well, again, that comes down to a lot of interaction with the customer. The best aptamer from our point of view is one that fulfills your need. So we will screen the aptamers as close to your end goal as possible. So throughout the seminar here, I've mentioned our Aptabind process, the protein purification. That is a key example. So we might say an aptamer, we've generated a fantastic aptamer that sticks to this target. That's great, but if it doesn't let go again, it isn't actually the best aptamer. So affinity is not always the best aptamer. It's not always the end goal. So we work with what you need as the end goal, and that's how we pick the best aptamer. Does it function as it needs to be?
Just picking up on the theme of affinity there, Dave, we have been asked the question, what's the typical sort of dissociation range or affinity that we achieve with aptamers?
Yeah, that's fine. It does depend a little bit on the nature of the targets. So a population of aptamers against a protein target, we would typically be aiming for low hundreds of nanomolar for the population. And generally, if we achieve that, it means that when we identify the individual aptamers, we will have affinities in the low double-digit, even single-digit nanomolar is typical. It's not unheard of if you have a good nucleic acid binding protein, you can have aptamers in the picomolar range. Small molecules, obviously, there's a bit less to get hold of with the aptamer, so the affinities tend to be a little bit lower, but generally they're in the sort of hundreds of nanomolar to micromolar range. We have had examples where we've generated small molecule aptamers, though, that are also down in the double-digit nanomolar range.
Just to remind people that online, you can ask a question live through the form on the website, aptamergroup.co.uk/questions. And we just have received one here. How do you introduce specific release conditions during selection if you want the aptamer to release the target?
Yeah, okay. So during our selection process, if specific release is not an end goal, we would essentially immobilize the aptamer target onto a surface and use that to recover the aptamers. We would then denature that whole system and release the aptamers, which allows us then to recover it. So that will release absolutely everything. If you have a specific release condition in mind, again, to come back to the aptabind situation, if you have specific elution conditions, we will actually incorporate those in our aptamer selection process so that only aptamers that bind under one set of conditions, but then let go again under your defined conditions, only those sequences are recovered and only those go through to the next round. And by doing that, we build in this need for defined binding and then release conditions. Another question that came up is somebody commented that on our website we actually don't mention the fact that we use next generation sequencing in our selection process and wondered why this is. So it's a very good question. It's true that you do see a lot of aptamer publications these days where they produce some lovely data showing next-gen sequencing and showing enrichment of sequences and their evolution throughout the process. We have looked at this in the past and we actually found that it can be quite misleading. What I mean by that is the aptamer selection process itself has a lot of built-in amplification steps. So you're doing PCR as part of the aptamer selection, and that in itself is going to enrich certain sequences. So if your population, for example, has a sequence that amplifies more readily than others, that can emerge in next-gen sequencing as a dominating sequence. And it can actually be quite misleading. As I said, you might see that and say, oh, there is a good aptamer sequence because it appeared in round 5 and has taken over my selection. And it's not an aptamer at all, it's just a sequence that happens to amplify very well. So what we tend to do is say, we're not going to be persuaded by sequence data. What we're actually going to do is screen based on the function that the customer needs. So we will isolate a plate full, a 96-well plate of aptamers, and screen them for function, and then determine the sequence of the best performing aptamers. This way we're not biased by what we think might be an emerging sequence. We're really looking at what is functional.
Okay, that's great, Dave. We're running out of time. I think we'll just run this for another 4 or 5 minutes maximum. We've had another question come through live. During the screen of a protein against your DNA library, could the best aptamer differ if the selection is made, for instance, in a complex sample, such as serum, instead of using purified buffer conditions? Before you answer this, I'd just like to say that this raises to my mind that we're often approached by people who have seen a potential Aptamer of use in the literature, and they often take that Aptamer and get it synthesized against their target of interest, and then they're occasionally disappointed in its utility because the selection process that would have been used to identify that Aptamer in literature is not in the environmental conditions of use that they are trying to develop one for their application. So I think that partly answers that question.
Yeah.
But yes, do you get different Aptamers if you select in buffer as compared to a complex matrix?
The simple answer to that is yes, you will. Almost certainly. The reason for that is that in a buffered system, you might introduce certain salts that would not necessarily be in a complex matrix like plasma. So you might find, for example, that if you put a little extra magnesium in there, you promote the interaction with the Aptamer and the target, where you wouldn't normally have seen that interaction in a magnesium-free situation. So yes, the buffer conditions that you do the selections and your screens in will definitely affect the outcome. What we tend to do, again, is start with the end goal in mind. So if we know that the customer wants to work with an Aptamer in plasma, but we've introduced some additional salts to promote that interaction, we will gradually phase the buffer out and phase the plasma in to the selections as we go to identify the Aptamers that have the best chance of working in the first situation, but then function as they are intended by the end of the selection process.
And back to the situation I described, if someone is interested in a literature Aptamer, we can do some work to evaluate its potential utility in the end situation.
Absolutely.
We can do some characterization and test for its potential use on a customer's behalf.
Yep.
I think we have 2 minutes left. We're getting towards the end of time. There's one more question I'll take off the list here. Going right back to the beginning of the process, how do we select the oligonucleotide pool for the SELECT process?
Yeah, it's a very good question. So we actually have a range of different Aptamer libraries in stock with different structural features, different random region lengths. We tend to pick them based on the customer's end goal, and the nature of the target. So, for example, a small molecule target is not going to need a long nucleic acid sequence to recognize it. So, for example, we wouldn't use something with a library with a 60 nucleotide random region for a very small target. So we tend to try and choose the Aptamer library based on the size of the target and an estimate of what's likely to be needed. We can also build in structural features if you know, for example, that your protein likes to bind to stem loops, for example. We do have libraries in stock that have an inherent stem loop structure. We can use those if necessary.
Okay, well, thank you very much, Dave. I think we've covered quite a bit there. A lot of information has been provided. I want to thank everyone who's registered and dialed in for their time. We hope this was of interest and useful to you. The webinar's been recorded, so you can watch it again or refer your colleagues to it for viewing. I want to thank my colleague here, Joel Sutcliffe, who set up the webinars, and he will contact you again soon with information about the next webinar on the 25th of October. For any further questions or any project-specific inquiries, if you want to contact the team here, there's the info@aptamergroup.co.uk email address, which is on the final slide there. The company phone number is there and available off the website. So it just remains for me to thank you all for joining us once again. Enjoy the rest of your day, and hopefully you'll be joining us soon, well, in about a month's time for the next webinar. Thank you very much. Goodbye.